Ayan, magandang araw sa ating lahat. So we are down to the last day of the 26th SINSM under the biology discipline. So I am Brian Alison Mortos, one of the instructors here at the Department of Biology in UP Baguio, and I will be your host for today since si Sir Dane ay may prior engagement. Ayan, magandang araw, magandang umaga sa ating lahat, sa mga nagagandahan at nagagwapuhan nating mga participants for today. I hope you already had your breakfast para naman may energy tayo na makinig dito sa magiging lecture natin for today. So sa mga hindi pa, magkape muna tayo kung wala pang na-prepare na breakfast si Mr. or si Mrs. or yung parents natin kung sila yung kasama natin sa bahay. Ayan. So I hope and I'm sure by now, marami na kayong natutunan from our previous speakers, Professor Easter Joy Sajo and Professor Madison Munar. So for today, of course, meron na naman tayong bagong matututunan under Professor January Pagaduan. So ayan. So just some few announcements before I give the floor to Sir Maverick Tamayo. About the certificates, kasi may mga nagtatanong na regarding the certificates. So the certificates, they will be given maybe a week after the last day, which is today. So yung certificate, ang criteria niya, dapat you have attended 2 out of the 3 lectures and kailangan na-evaluate nyo yung speaker. So yung evaluation ay binibigay naman every after ni speaker, every after mag-lecture ni speaker. So ayan! So kung sana lahat tayo ay maging okay for today, maging maayos yung magiging flow ng ating program for today. Unfortunately, this is the last day, pero I'm sure marami naman na kayong natutunan from our previous speakers and marami pa kayong matututunan sa ating speaker later. Ayan. So, ngayon, I will be giving the floor to Sir Maverick Tamayo, also an instructor here at the Department of Biology in UP Baguio. Sir Mavs, take it away.
Hello, Sir Brian. Thank you very much for heading the introductory portion of our today's last day of SINSM, no? Ayan, so narinig na ba ako or baka ako lang nakakarinig sa sarili ko? Pa-thumbs up naman po tayo dyan kung narinig po ako. Okay lang ba? Narinig po? Alright. Sige po. Maraming salamat po. Okay, so hello mga ma'am, sir. Kamusta po tayong lahat? Welcome po sa uling araw ng ating pag-aayuno. Joke lang. Day 3 ng ating 26 SINSM. Kamusta po tayong lahat? Nag-almusal na po ba tayo? Nagkape siguro? Nagpandesal? Nag-jogging o nag-ehersisyo na po ba? Dahil ngayong araw ay amin pong babanatin ang inyong mga buto at kasukasuan. Joke lang. So yun nga, maligayang pagbabalik po sa ating Zoom platform. Minsan ko lang po magawa ito, no, sa aking mga kapwa teacher, kaya ako po ay magpaparecite. Ayan, no? So siguro, kung lang po, kaunting recap or gist po, no, or kung ano po yung natutunan natin during yung ating day 1 at saka day 2 ng ating SINSM. So short essay lamang po tayo or short reflection ng ating gustong sabihin at ibahagi sa ating mga kapwa mga guro, no? So akin pong tatawagin ang pinagpalang dalawang participant po, isa sa day 1, isa sa day 2 sa aking mahiwagang tambiolo. Okay. So sino ang maswerteng mananalo ng trip to Jerusalem? Okay. Pwede po ba nating tawagin si Ma'am Joanna Rose Panergo? Ayan. Diyan na po ba tayo, ma'am? Hello po, Ma'am Joanna Rose. Pakigalaw po ang baso. No? Okay. Wawawin po ito. Nanalo po kayo ng washing machine. Okay. Mukhang nahirapan pa mag-connect sa audio si Ma'am Joanna. So next tayong bunot. Okay. Si Sir Stephen. Yan. Stephen. I-ask to unmute ko na lang siya. Ayan.
Hi, sir. I couldn't hear your voice well. Can you
Ay,
speak?
hello sir, hello po.
Yeah.
Narinig na po ba ako?
Mm, yep.
Okay po. Sir, one lang po. Gusto ko po sana magkaroon po tayo ng konting recap, no? Mula sa ating day 1, from day 1 to day 2. So, ayun po. Kayo po ang mahiwagang nabunot sa aking tampiolo. Konting short reflection lang po, sir, regarding po kung anong natutunan natin dun sa ating day 1.
For day 1, the use of Labster, it's really kasi
Mm-hmm.
we're also using it in our setup right now, blended setup. Labster, and also yesterday was Blast, phylogenetic
Yeah.
analysis. I think if we could like somehow uh, look upon different aside from Labster and Blast, and we could somehow develop virtual simulations for the students use. Yun po yung recommendation ko, kasi limited for, let's say for example Labster, like it requires like number of students to for them to avail the discounted price. And for Blast din, it's very hard for like students that are living in remote areas kasi they couldn't access it, especially NCBI, GenBank, and many more, many softwares.
Mm-hmm. Okay. Sir, salamat po. Tama nga po, no? Yun po talaga yung napakatindi at napakalaking problema po natin. Especially, though gusto nating i-educate more at i-share sa mga students natin yung experience ng virtual setup. I mean, yung virtual laboratory, no? Talagang problema po most specifically sa ating gadget at internet connection po, no? So, ayun po yung hopefully, no, ma-solve natin sa mga susunod na mga chances na magtuturo po tayo ng ating virtual or online setup. Okay po. How about for day 2, Ma'am Marie Nicole Bugayong? Ayan po. Hello, ma'am. Good morning po. Hello, ma'am. Pakigalaw din po ang baso. Joke lang. Hi, ma'am. Okay. Ayan po. Naku. Para mga estudyante rin, no? Pag nagpaparesearch si teacher, natatakot. Tumitingin sa baba. Ayan. Ay, halon. Hello,
Morning
ma'am.
po.
Ayan po. Morning, ma'am.
Nung dito po, ang mga key takeaways ko po ay yung dalawang sites or software po na pinakita ni Professor. Yung first po is yung Blast by NLM ata po, and the second is MegaX. So malaking tulong po na shinare po yun kahapon dahil may papakita po namin sa mga students yung mga pwedeng maging potential research po nila na pwedeng magamit na makakatulong po. Ayan po.
Ayun. Alright. Maraming salamat, ma'am. No, sana turo natin or ma-share natin po ng maayos, no, yung atin pong mga napulot na aral during the first and the second day of our SINSM. Okay po, maraming salamat po. So welcome again sa ating day 3 of the biology discipline. 26 SINSM, genetics and cell biology at the crossroads of different biological disciplines. Okay, so simulan po natin ngayon ang ating day 3 sa pag-i-introduce ng ating resource speaker. So may I please introduce to you today's speaker, one of our resident genetics instructor in the department. Ayan. Yaman din lang at ang topic natin for day 3 ay teaching genetics in an online setup. All right. So Mr. January Pagaduan is a junior faculty at the Department of Biology, College of Science, University of the Philippines, Baguio. He earned his Bachelor of Science degree in Biology, graduating as cum laude at UP Baguio in 2015. Currently, he is a candidate for the Master of Science in Conservation and Restoration Ecology program in the same university. He has been actively participating in his institution's researches centering on biodiversity conservation and ecological restoration in the Cordillera region. As a young and passionate faculty researcher, he aims to continue his academic endeavors and carve his niche on restoration ecology, particularly on the field of environmental microbiology. So before we start today's knowledge and fun-filled lecture, may I please again remind everyone to please turn off your mic and post your questions sa ating pong chat box for our question and answer portion later. Also, please do not forget to answer the evaluation form for our day 3. The link for that will be posted po dito sa ating Zoom and social platforms accompanied by a distinct QR code. So without further interruption, Sir Geno, please take over the screen sharing. Thank you very much.
Okay, thank you very much, Sir Brian and Sir Mavs for the introduction. Sir Mavs, thank you for the generous introduction. Okay, so hello, good morning. Before I start, first, I just want to express or I just want to tell you guys that I am really loving this crowd. For the first 2 days, I've been supervising the activity and natutuwa ako dun sa general atmosphere, attitude ng participants. Yan, so pagka may nagtanong and for example, ano po yung ginagamit na platform at alam naman ng participant siya nang sasagot. So essentially you're helping your colleague, your co-attendee. And kapag ka nag-good morning, nakikita ko ang dami nag-good morning, good afternoon, or kapag ka tinanong kung ano yung almusal, sumasagot, kung ano yung kinain ka ng tanghali, sumasagot. It's really warm. And I hope mag-continue yung ganito especially sa atin, no, among teachers. Okay, so yun. Thank you. I'm seeing hearts right now. Yan, so thank you very much. Yan, so for today will be day 3, the last day of our activity, and I hope we will finish strong. And for my talk, I'll be delving into the pedagogy. No? So the first 2 days, may teaching instrument na pwedeng gamitin, and research. Yung sa akin, mas mag-focus po ako on the teaching side. No? Not just on genetics, but I think by extension, some of the things that I would mention are also applicable in some lab-based or at least subjects with lab component. And so with that, let me just share my screen. Ay, mali. Okay. Oops. Ayan, nakikita niyo na po ba yung aking screen? You should be seeing my title slide. And teaching genetics in an online setup. Okay na po ba, Mr. Moderator?
Yes, sir. Opo.
Okay. So, as I mentioned before, for today, this will be my topic, teaching genetics in an online setup. Pasensya na, walang kaamor-amor yung aking title. I don't have the time to sexify my title. So, I'll just daanin ko na lang sa content. Okay. So, for this presentation, and this will be our agenda for today. So general concepts, list of concepts, or the roadmap, how I will be discussing my presentation. So first, I will be discussing the challenges that are commonly encountered in an online setup in general, muna. And of course, I'll also share with you the strategies, suggested strategies naman. We're not imposing. Parang based on our experience, these are some of the things that maybe we can use. Yan. Actually, dito pa lang pwede nyo ng take away na to. No? So for the challenges, yan, poor connection, assessment delays, kinesthetic limitations. We will delve into these topics in a little while. Then for the strategies, ayan, utilize open educational resources as well as other available resources. Next, the power of pictures, biological simulations, affiliative approach, in garage experiments or home experiments. Yan. So kung mapapansin niyo, I also crafted this in a way na
hopefully makatulong sa atin to remember these things better. Yan. Okay. Okay? Okay. So for the challenges, basically we will be discussing the barriers to effective online learning. And for the strategies, as I've mentioned before, these are our suggestions based on our experience. Okay, so challenges. But before we go into the challenges na I will be mentioning, first, we would love to hear from you. We would love to hear you out. Okay, so grabe naman po yung day 3, ang daming pa-activity. Yes po, ganun talaga, oo. We really want it to be interactive. So before we delve into the content of my presentation, may konting activity lang po tayo, magpapa-activity lang ako. Please, yan. For those who are able, hindi rin naman po to required, but for those who are able to do this, please, may I enjoin everyone na walang limitation sa kanilang bandwidth to join this activity. This is very short, and actually you can also employ this in your classes then. So please go to this website, www.menti.com, and use the code shown here. Okay, so when you go to the website, you should be able to see this interface, itong puti yung background. So very simple lang yung interface pagkadating nyo doon sa may site na to. And then you just need to enter this code. Okay. May I request the tech team and secretariat pwedeng pa-furnish nung code sa chat box so that they can just easily copy and paste it. Yeah. And baka kasi I will remove my share screen in a while. Yan, so basically kapag dating nyo dun sa site, ito yung makikita ninyo, you will enter the code and it will prompt you to answer a question. And this, you should see this tawag dito, this screen, ganitong screen, and then you need to answer. So for this activity, maybe let's have 3 minutes, 3 minutes, and then after this we will try to see yung feedback ninyo. Okay, I'll just stop my screen share muna. Yan. So nakuha po ba? Yan, menti.com and then the code. Yan. So sige. So habang habang ginagawa niyo to, dadaldal lang ako ng konti. I also want to share that most of the things that I'll be sharing ay produkto ng mga natutunan din namin sa mga workshops na pinagdaanan namin. Especially, 'di ba, it's a difficult time para sa ating lahat, na biglang nag-shift tayo sa online learning, especially sa mga teachers natin na sanay na dun sa traditional method, and then biglaang may shift, and napakahirap talaga, lahat tayo nabigla, and so lahat tayo nangangapa. Yan, so because of that problem, we had series of workshops, and I tried to distill all of the things that I have learned into this presentation. So some things dito na makikita ninyo, baka they're not new, baka nakita nyo na-present na siya sa ibang conferences. That is because I'm trying to distill yung mga learnings ko din naman and share it again. And so especially galing kay Ma'am Prof. Hervasio of UPOU, Prof. Florito of UST, and Prof. Padilla of UP College of Education. And sa kanyang isang talk na course pack ganern. Okay, so I think we have sufficient naman na tawag dito na results. Okay, yan. Keep them coming. Okay, let me just reload. Ayan. Okay. Okay, so I think we've gathered enough. Let me just share yung result. Ayan, so you should be seeing my screen now. Ayan. Oo. Ito yung inyong responses. Ayan, nakikita po ba? Ayan. So for this activity, you can also employ this in your classes. This is mentimeter.com. May mga paid versions, but the good news is meron yung free version for the basic presentations lang. So for example, I'm using the free one, and ang isang pwedeng gamitin is the word cloud. No? Word cloud. So same sa activity na ginagawa ninyo ngayon. And ang gagawin niya is it will place your answers into this graphic. No? And para dun sa mga nauulit-ulit na sagot, they actually appear bigger, no? Appear bigger in this word cloud. So somehow, parang visually, makikita ninyo ano yung mga common na problems. Although ano din kasi, I think medyo sensitive siya dun sa wording, no? Yan. Pero yan, makikita ninyo ano yung mga shared na problems natin. Ayan, nangingibabaw. Poor connection, poor internet connection. Connection, yes. Unstable internet. Ayan. Difficult to teach, I agree. Ibang-iba yung pedagogy kapag ka-online ang setup. Sige, let's just review some more. Active participation, yes, yung engagement ng students, it's quite hard, no, to engage them into the activities sa kapag ka-online setup. Lack of devices, yes, and resources, that's also true. Low digital proficiency, yes, in terms of digital literacy, this is true both on the side of the instructor and the side of the student. No? Hindi lahat tayo ay biniyayaan ng circumstansya na may access sa devices. Yung iba sa atin nalilito pa, hindi tayo pare-parehas. Yung iba may head start when it comes to online learning. Yan, student engagement, yan. Ano to? Charmander. Okay, sige. Pogi. Thank you. Power outage, yes. Totoo din yun. Especially ngayon kapag medyo summer ata, napapadalas ang power interruption. Checking lab exercises, yes. Especially kapag ka-lab component, it's extra challenging in an online setup. At least yung lecture, since concepts siya, you can deliver it through the presentations lang, no? Pero kapag kayong sa lab, it's really strategy. Difficult to teach, nabasa ko na yon. Limited resources, yes. Problem with lab works. Totoo yan. Okay, so makikita natin, and I'll just stop sharing and then balik tayo dun sa aking presentation. Ayan. So thank you. No, thank you for participating in this short activity. So makikita natin may mga recurring problems or may mga common problems. And I know yung iba sa inyo nakaka-relate sa isa't isa na ito yung mga common problems na na-encounter natin in this online setup. And I'm not here to give you solutions but suggestions. Kasi yung iba, some of the things that I would mention might work in the context of, in some contexts, but not in others. And so hopefully, may mapulot din kayo dito, and hopefully may maibigay din kayo. Later on, I'll also have another short activity, no, na i-discuss natin in a while. Okay, so thank you very much. Yan, we love hearing you out. Merong nakikinig sa inyo. Kumbaga may mga accountability partners tayo, hindi lang tayo namumroblema dito, no? Pati pala yung kapit bahay kong school, ganun yung problema. And yes, may common grounds, kumbaga. And somehow it's comforting, no? Na hindi lang pala kayo may ganitong problema. Okay, so now let's proceed. Yan. So most of the things that you've mentioned are also also in these points, Pero ito lang, I'll also share our own head scratches and sighs. May mga frustrations din kami pagdating sa online teaching. So summary, yan, poor connection, assessment delays, and the kinesthetic limitation. So let's just try to expound further. No? So first, poor connection. Poor connection, especially internet. On the technological side, there's a poor connection. From time to time, yan, may mga estudyante tayong or teachers din na limited ang bandwidth, intermittent ang connection, nawawala, humangin lang ng konti, umulan lang sa labas, nawala na po yung internet ko. And that really affects an online setup. Kasi internet ang kailangan mo for an online setup and then biglang mawawala. That's really a problem. But poor connection would also refer to poor connection socially and emotionally. In this online setup, we don't bond with our students as we bond with them before. Because of this online setup, may mga elements na strip off. And because of that, it resulted to this, no, to poor connection with our students and the connection of students to us. And because of that, we are blind, no? We are blind to their situation. Hindi natin alam kung ano eksaktong nangyayari sa kanila. We are alienated from students' perspective. Hindi natin nakikita na sa bahay nila, blurred na yung pagiging estudyante nila, role nila bilang estudyante, at role nila bilang anak. Or role nila as breadwinner, kung breadwinner man sila. And hindi natin nakikita yon because of this poor connection. And the other direction is also true. Hindi rin nakikita na ng mga estudyante kung ano nangyayari sa atin na teachers. No? And because of that, because of this poor connection, it often results to miscommunication and mismanagement of expectations. So what are we trying to say here? There's a discrepancy on the expectation and reality. So kunwari si teacher, I would draft this activity, it's good for 1 week. Ganun. I think it's doable. Ganun. If I'm just using my perspective, it's doable in 1 week. Okay. However, hindi natin nakikita, pwedeng dun sa side ng estudyante, may ibang factors na mangangain ng oras nila, hindi lang yung ating activity. For example, hindi lang yung subject ko, may subject pang iba. Paano kapag other instructors, teachers would think the same? Kawawa din yung estudyante. On the side din ng teacher, parang students would also like, bakit hindi pa binabalik ni ma'am tsaka ni sir yung quiz ko, Diyos ko po, ganun. Hindi rin nila nakikita na on our side, it's also difficult to create, activities, to plan. Yung think tank natin, overused, no? So barely we have energy for the checking. Kaya may mga ganong dynamics, no? Na kapag if you're blind to those dynamics, ang dali na lang frustrate at magalit. Okay, so yan, miscommunication, mismanagement of expectations. So what's the resolution, or not really resolution, or what can be done? Yan. It is, we need to understand the learner's context. We need to be aware of their limitations, psychological, financial, technical. Nabanggit kanina, it's about digital literacy. Not all of our students would have the technological proficiency. Yan. Do they know how to log in, participate in classes, submit work, and communicate with teachers and their classmates? Kailangan somehow meron tayong idea, no? Idea kung dun sa level ng digital literacy ng ating students. Also, on the side of the teacher, digital literacy din ng teacher. Parang kahit ako din, relatively bata-bata, believe me, I'm also, nagpapanik din po ako sa mga ganito. No? Ang daming lumalabas sa screens, ano yung nagpapakita sa screen, ano yung wala sa screen. So yun. So, understanding, no? Each other. That's important. No? As for the internet connection, wala po akong may i-offer na resolution for that. Wala po akong pambayad ng internet. Opo, mukhang kailangan na nating i-built in din sa ating budget. Anyways, next, assessment delays. So here, just to give a description muna, it's about the delay in giving and receiving feedback. With an online setup, I think most of us would agree, there's a lot of friction in giving an assessment or evaluation form. Kung nung face-to-face lang ay pwedeng, kunwari, may activity, okay, mali yan, ganyan, okay, or palitan natin yan, ganyan, or mas maganda ata pagka ganito, nasabi mo na, tapos na. But for online setup, just ko po, i-articulate ko pa yan lahat sa email. Greetings of peace and prosperity. I am writing this letter, ganun, with the intent of, ganun pa, no? Parang, that in itself is a friction. And compounded, when those sources of friction are compounded, ang hirap nang magbigay ng feedback. No? A simple comment would take a lot of time and effort to articulate. Yan, so tsaka ang isa pang problema is that there's a complication of being unclear and misunderstood kapag nagbibigay ng feedback. Yan, so ganon. Okay, paano ba? Example, ayan, example. So, nag-submit si student ng output, na-receive ko na sa email or na-receive ko na dun sa platform. In our institution is the UP-VLE, meron kaming platform. Na-receive ko doon, ita-download ko, i-sort ko pa siya, and then tsaka ko siya i-check. Ang daming added steps, ang daming friction. Kung sa online sana, okay, sinabit ni student yung printed, check, okay, balik. Yan. Another problem is that yun nga, dahil ang communication, you strip off the non-verbal side of the communication, it's prone to miscommunication, misunderstanding. Okay, so this lag in giving and receiving feedback, ano yung isang major result? Loss of engagement with students. Pwedeng ma-feel din ni student na, okay, am I just submitting for the sake of submitting? I thought this was an avenue for learning. And they would feel that they are learning if they are receiving feedback. No? If they are receiving feedback. Okay, so yun. As for the resolution or suggestion for this is we need to set up systems for effective communication. So teachers, kapwa teachers, educators, it's only natural that we can't reply and give assessments as frequent as we want to. No? But to help us, we would like or we would want to set systems to effectively communicate our feedbacks. So how do we do this? One of the suggestions that I can give is to use several modalities, no? Several modalities in communicating with our students, but not too many. So sa akin, experience ko, I would have FB groups for my students, I would have email threads, and dati gumamit din ako ng Google Drive. Yan. Tapos doon ko nilalagay yung mga activities. Ganun. So we would want to have multiple modalities in communicating with them kasi iba-iba, depende doon sa goal mo. For example, FB and email. Those two modalities for communication or those two platforms, essentially they do the same thing. They can deliver messages. No? They can deliver messages. It's just that magkaiba sila ng context. So for quick exchanges, quick informal casual exchanges, you may use FB, GC, kahit GC lang. But for sending important files, ganun, or yung mga kailangan ng paper trails na communication, then use your email. Para hindi ka laging greetings. This is to inform you that hindi ganun. Pwedeng sa students, please check pwedeng ganun na lang sa GC. Yan. So several modalities. Kung saan mas madali. Actually, depende pa rin sa context ninyo. Some of my colleagues would use Discord, yan, as their communication tool. We want several modalities but not too many kasi kapag sobrang dami naman, mahihirapan, nakakapagod, nakaka-exhaust. No? You need to check a lot of apps para lang makita yung messages. Baka may mga estudyante nag-message sa email, sa FB, sa Discord, sa Instagram, or you name it. Kaya yun, we would want to have several modalities but not too many. Okay, next. Kinesthetic limitations. I think this is just one of the pinaka-obvious at pinaka-challenging talaga sa lahat. no, kinesthetic limitations. When we say kinesthetics, we're referring to those that would require the sense of touch, yan, proprioception. Yan. So for example, ipapalpate or itatouch yung specimen. Ayun. With this problem, there's a restricted haptic element of the activities. Yan. Pag haptic perception of objects by touch, yan, proprioception. Yan. So yung means of conveying information through physical contact is limited in an online setup. Yan. So this is because of yan nga, you can only do so much virtually. Tapos may students and some teachers would be have restricted access to research spaces and resources. Without those important resources, may mga activities tayong hindi magawa. Yan. So, ano yung effect? Compromised practical skills. Yan. Since you strip off the haptic or the kinesthetic component, some learning objectives are compromised, especially those that will need practice or fine-tuning the motor skills. Yan. So motor skills, we're not just referring to exercises. No? We're also referring to, kunwari, in genetics, yan, sa paggawa ng slides ng allium root tip. Yan. Hindi natin maituturo yung kung gaano kalakas yung pressure na i-apply mo when you would press the onion root tip, or kung paano i-refine yung motor skills mo in slicing, no? Or in preparing slides of our specimens. Hindi natin yon matuturo online. Yan. So as for the advice, suggestion, or resolution, take stock, not think I'm stuck. Ano yung gusto nating sabihin dito? First, recognize that there are some activities that just can't be performed. There are learning activities that just can't be performed. So ang discouraging naman po nun. No, not to be discouraged. We need to recognize yung mga pwede nating gawin at hindi natin pwedeng gawin, to know which teaching dilemmas or which problems are out of our control and which problems are workable. Okay? No. It's because we have limited bandwidth, psychological bandwidth. Our resource, our energy is a limited resource. So we would want to allocate our energy on those that we can work on. Kasi yun yung pwede tayong makapag-provide ng solutions. Okay? Okay. So, yan. To demand accountability, no, to those who are in position is a noble cause. But we also need to be responsible on our microcosms, on our own spheres. No? Yan. So, idagdag ko lang ito, medyo konting ano ba tawag dito? Sidebar. And di ba sabi, with great power comes, you can type it in the chat box. Yan. With great power comes and great responsibility. Tama? Sabi ni Uncle sa Spider-Man. With great power comes great responsibility. However, the reverse is also true. With great responsibility comes great power. No? If we take responsibility to some aspects, no? If we take over to things that are workable, it empowers us. It empowers us. It makes us think of the strategies that we can so that we can move forward. Tapos gusto ko yung nabanggit ni Sir Don dun sa beginning na ano nga ba yun? Yung mother of reinvention. Necessity is the mother of reinvention. And yung necessity na yun, it's driven by crisis. Now we have crisis. And hopefully, by taking over on the things that we can control, we can move forward. Okay, so next, ayan, let's go to our strategy, some of the things that we're employing para somehow naman ma-resolve yung mga na-encounter naming concerns. Yan, so our strategies, yan, summary, yan. First, UP bag, no? Utilize available resources, the power of pictures, biological simulations, affiliative approach, and then garage experiments. Okay, so first, let's have this, yan. Utilizing available resources. Yan. So, online, access to the internet, no? Yun yung isang basic na kailangan nating gawin, or we're very much acquainted with the internet because of this online setup. And the internet is a cornucopia of information. Ibig sabihin, it's teeming with information. It's a blessing and a curse, actually. No, both good and bad. Kasi it's filled with information, both good information and bad information. Yan. So we need to filter through. No, we need to filter through. Yan. So how can we fully utilize available resources? Of course, we can use good old search engines. You Google them, marami nang lalabas na results. Yan. So, and we can borrow resources actually. We just need to attribute them properly. No, give the citation at saka credits. Hindi po credits to the owner ha, meron tayong rules in giving credits to those who own the material. Okay, so another thing is this. We can take advantage of open educational resources, or OERs. There are a lot of sites for this. Open educational resources, usually pwede siyang gamitin, na? Pwede siyang gamitin. Yung mga resources na nandito ay pwedeng gamitin sa presentations, although yung license niya nag-iiba-iba. Some, it permits editing of the, kunwari picture, editing of the picture that you have retrieved, or yung iba pwedeng just for display only. Yan. So, as I mentioned, madaming sites, but for me, personally, these are some of the sites that I found useful. So these are just suggestions. We have OER Commons. Actually, I have the link here. Kung makikita ko, if I hover my mouse here, makikita ninyo dyan yung link. I can provide all of the links to this later. Yan. Then Creative Commons. Yan. So we have this. You may want to visit these sites in your free time. Okay, so as I mentioned before, the internet is teeming with information, both good and the bad. So it all boils down. So kapag may topic ka, most probably meron kang mahahanap. May mga gaps lang, pero meron kang makikitang resources. But because internet is diluted with information, it all boils down to effective curation of resources. Parang you determine which resource would work and which resource is not effective in my context. And maganda if they are sharing like this. For example, I can share ano yung mga nag-work sa context ko, ano yung hindi nag-work sa context ko, and maybe your colleagues would also learn from your experience. And so sana collaborative din, no? Or we help each other in curating these resources so that we just filter and get the high quality ones. Yan, so ito, I'm lifting this from the workshop of Prof. Padilla. There's no need to reinvent the wheel. Yun din. In a certain topic, most probably, there are a lot of resources there. You don't need to create another resource. You can. It's good. However, if it stresses you so much, then just find a good resource attribute and then just put it into a new packaging. So we should be standing on the shoulder of the giants. Ito yung isa sa mga metaphors, no, ni Sir Isaac Newton. Parang this would be a metaphor. That would mean we use the understanding gained by major thinkers, gained by the giants, who have gone before in order to make intellectual progress. We build on the resources, on the information na available na. So from the resources available in the internet, we just look at them, digest them, make a new packaging, yun yung isend natin sa students. Kasi I know makaka-relate po kayo, ang hirap maggumawa ng module from scratch. From scratch. And yung lahat ay galing sa'yo. So one of the strategies is to look for good resources, and then compile them or provide a new packaging. Okay, next, power of pictures. Ayan, so isa sa mga challenges ng online learning is that we have restricted access to research spaces, to resources. So walang microscope, walang reagents. Paano yun ginagawa po namin yun sa aming lab activities? So ang isa sa mga ginagawa namin na adjustment ay we utilize the power of pictures. No? So, di ba? A picture is worth 1,000 words and is also sure a worth of thousands of data. Depende dun sa paano mo gagamitin yung picture, marami kang pwedeng makuha actually. Or may mga learning activities kang pwedeng or learning outcomes na ma-achieve. Yan. So basically, ang gusto kong sabihin dito is we're actually using images of slides as substitute for microscopic examinations. Just for the microscopic examinations, not for the real skills. Kasi yun nga, as I mentioned before, wala tayong magagawa doon. Pwede siguro kung sa garage experiments or home experiments. However, in this case, we can utilize micrographs, no, to achieve at least ito, yung learning objective na maging familiar yung students sa ilang microscopic structures, kung ano yung mga makikita sa slides. Yan. So for example, ito, meron po bang makakahula kung ano ito? Ayan. Ano po ito? Ano kaya ito? Mitosis. Yes. Okay, very good. Okay. Okay, yeah, very good. So dito ang ganda, no? Pagka sa lab ito, ang hirap gawin. Ang hirap i-achieve yung ganito kagandang slide. But anyways, ayan, at least we have the benefit of yan, online learning. We can use pictures, just put proper attribution, or kung license naman siya to free to use, okay lang, no? So ayan, so this is a slide of onion root tip, no? Onion root tip with stains na nagpapakita ng stages ng mitosis. So these are actively dividing cells. So remember, sa may onion root tip, no, meristematic, it's actively dividing. So expected, makikita mo dyan, you have cells that would undergo mitosis. And so for example, you provide this picture to students, or you ask them what are the ayan, just thinking on the top of my head, what are the stages of mitosis that you can see here? Or given that this is an onion root tip cell, what is the biological process that's prominent here? Kunwari ganon. O mapapaisip sila, onion root tip, actively dividing, okay, mitosis. It should be mitosis, not meiosis, because it's a root tip, no? Somatic cell, not a germ cell. So most probably it's mitosis. Or for example, pwede na itong gamitin for mitotic index, no? Pag-solve ng mitotic index. Ano pa, or ipalabel sa kanila kung alin dyan yung anaphase, telophase, ano yung prophase, ano yung metaphase. Sorry, wala ko sa order. Prophase, metaphase, anaphase, telophase. Yan, so things like that. No? Yan. Mga ganong tanong. Okay, so ito, just to share, yan sa amin, ayan, nakita nyo na ba yung slide? Ayan, ito ay slides po namin ito. Ayan, so, ang tawag dito? These are also allium root tip, no, cells stained with aceto or sane. So pwedeng ganito. Actually, this is one of the activities that we've given our students. We gave them these slides, and we just had research questions. So dito, yan, pinamitotic index namin sila. Sir, ang hirap naman nun, manual nilang titignan. Ganun din naman po nung face-to-face. Para ma-feel nyo pa rin po. Para not to, ang tawag dito, para hindi rin sila ma-shortchange. No? May mga ganito. This is to train their eyes also. Diba? And so for example, ito din, ayan, grasshopper testis at 400, yan, ganyan. So ano dito yung mga, of course, limitations din. But this is okay na yung ganito, no? Pati din naman sa actual, no? Hindi naman sobrang perfect din yung nakikita nating slides. Yan. So kunwari, you provide them this slide and then ask them ano yung mga nakikita yung stages of mitosis. Meiosis, since this is grasshopper testis. Yan. Sorry. Ayan. So, hindi pwedeng pagsabay ng laser pointer. So, ano? Ayan. So ano po yung mga nakikita nyo dyan? Ayan, you comment below. Ayan. Tignan ninyo. Oo. Okay lang din naman po graded. Ano yung mga nakikita ninyo? Or baka may mga na-imagine kita, oh, ito, ano to? Mga things like that. You can do things like that. And so kung wala pong access, pwedeng yon si teacher, siya yung gagawa nung slides kung may access siya sa lab and then send the pictures to the students. Or kung wala, we can always go back to the open educational resources. There are available pictures din naman in the internet. Although by experience, meron din talaga yung mga wala. But most of it, nasa internet. Okay, next, we have biological simulations. Yan. So, and we're acquainted with this. No? Nung kay Ma'am EJ, pati ako na in awe nung nakita ko yung sa Labster. Oh, ang ganda. With the music and all. Yan. Gamification, di ba? Gamification of activities. Yan. So, we can use biological simulations. So just to mention the nerdy stuff, pag sinabi nating simulation, it's a technique of studying and analyzing behavior of real-world system or process by mimicking it on a computer application. Yan. So, ang tawag dito, this is done through virtual labs. Actually, meron ding mga open virtual labs, meaning you don't need subscriptions. Although, compared to those paid virtual laboratories, virtual labs, mas maganda yung mga paid virtual labs, of course. So kapag kami mga free virtual labs, we manage our expectations. These virtual labs are, I think, product ng extension services na ibang universities. Yung iba, hobbyists. They're just hobbyists and enthusiasts. And so also, we can do in silico experiments. So this, actually, pag sinabi nating in silico, pag sinabi nating in silico, these would be those experiments or methods that are done using computational approaches done in a computer. No? So this is just to parallel, pseudo-Latin. This is just to parallel with the more common terms in vivo and in vitro. Diba? In vitro within the glass, in the test tube experiment, and in vivo within the test organism. And ngayon, mukhang mas sisikat itong term na to in silico. We have experiments that would be done in a computer, no, computational analysis. Ganyan. In silico kasi 'di ba, ano yung main component ng microchip ng computers? Silicon. Yeah. Okay, so these are some of, in relation to genetics, these are some virtual labs or sites, no, with resources that I found useful. That we found useful, sorry. And so we have the Fly Lab. Ayan, hindi na po sila pwede nang i-mate yung Drosophila, no? Hindi na nila kailangan magdikdik ng kamote at lagyan ng propionic acid saka ng yung pampaalsa, yeast, no? And then they would manually, although exciting sana, but in this case, hindi natin pwedeng gamitin yung mga nakikita po sa yung dumadapo sa banana peels. Hindi po yun kasi genetically hindi natin alam kung ano yung composition nila. Anyways, we have these labs. We have the PopGen Fish Pond, and ito, Learn Genetics sa, I think, University of Utah. This is also a good site, no, for resources. Ayan. Okay. Next. Ayan. Affiliative approach. Ayan. Actually, nung una, this is collaborative approach, but I need to fit it to the UP bag acronym. Anyways, affiliative approach. So this is also one of the things that we have observed. It's very important that we work collaboratively. So what's the common adage in relation to that? Diba? Teamwork divides the effort but multiplies the effect. Kapag nagkatulong-tulong tayong educators, no, pwedeng mapagaan natin yung trabaho ng isa't isa. No? Baka, for example, sa curation mismo ng resources, ayan, na-try ko yan, mukhang medyo mahirap, hindi user-friendly. Or ito, mas malinis yung interface, baka pwede mong ito. Yan. So, sa experience namin, it's ang laking tulong na dinadaldal ko yung mga ganito with my colleagues. Kasi may mga nasasabi sila, wait lang, parang may na-encounter ako. Especially, yan, shout out to Sir Ronnie, yan, Sir Ronnie Kalugay, for mentioning the Learn Genetics resource of University of Utah. It's really a good site for resources for teaching genetics. Ayun. Or may mga discussions, may mga answers na lumalabas sa discussions, no? Like parang coffee break, nag-uusap lang kayo. Oh, that's a nice solution. I think that's good. Ayan. So yan, we share experiences and best practices. So in this, maganda if we have a centered database, tama? Wouldn't you agree? Kapag meron tayong database nakalagay doon. And as an exercise, I would want you to establish databases. When we say it doesn't need, pag sinabi mong database, parang ang naiisip mo, it should be like thousands of, hundreds of thousands worth na database, na software. No, even a simple shared Google spreadsheet will do. No? Kasi ang kailangan mo lang naman ay compilation ng resources na accessible sa lahat. So in line with that, I actually tried to make this compilation of resources, and sige nga, I think I'll try to show that now. Just give me a moment. I-open natin. Okay. So I'll just share my screen again. Already nawala. Okay. Yan. So I just tried to do this. Ginawa ko lang siya para nag-set up lang ako ng system. But actually it's for practice purposes only, or not. Pwede niyo pa ring balikan to. So I created this Google spreadsheet. Yan. So I just put here the resources na pwedeng nating gamitin. Ayan. So the Mentimeter, the Menti, yung ginamit natin kanina, brief description para for the guidance na din ng ibang mag-access ng spreadsheet, subject tag, so that actually pwede kasi nating i-sort based on what column. So for example, na-populate na siya, and then you need to na-populate na siya, and then we need to look for just resources on kunwari biglang may naglagay ng vertebrate anatomy, pwedeng, and you want to compile them, pwede nating i-sort. I-copy nyo yung table ito a different spreadsheet, and then sort by column E, which is subject tag, things like that. No? So this is, all of these are just on the top of my head. I just tried to do them last night. No? And ito, maganda if we have things like this, yan, yun din mga mini-mention ko kaninang links, andi dito na rin siya. And I also edited the privacy settings so that everyone with a link would have access to this spreadsheet. And I would want to share this now. Would that be possible? Sige, I think I can, nakikita nyo naman po, nakikita nyo naman po yung, baka ang dami ko nang nadaldal, niyo pala nakikita yung spreadsheet. Nakikita po? And sige, tingnan nga natin kung may feedback. Ayan, delayed feedback. Visible po. Okay, thank you. Yeah, thank you ha. Kailangan din yung feedback. Oo, yung feedback. I-remind nyo rin yung students, kailangan yung feedback. Kasi that would prompt me ano yung kailangan kong gawin. Yan. Encourage them to give feedbacks. It also fosters interaction. Okay, so paano kaya ito? I'll just try to post the link. Ayan, sorry, everyone in the waiting room, everyone in the meeting. Ayan. So, I posted the link sa my chat box, and later on, those in the YouTube, pwedeng ibigay din namin yung link. Pwede nyo ring tingnan sa my Facebook page. Ayan, so ang bilis. Meron na pong, nakikita na natin kung sino yung mga andi dito na agad. No? So, dun sa mga nasa may spreadsheet na, is it accessible or do you have problems accessing it? Oh, kasi baka may mga privacy setting issues. Ayan. Ang bilis. Meron na mga 12 na ata agad ito. Ayan, so you can just access these links, no? And I edited the privacy settings to make everyone an editor so that you can also add. No? Kapag may nahanap kayong useful na link, you can also add. No? You can also add. Put it here. Yan. Just kunwari, may gusto kayong i-share, wag po tayong madamot. Kung may nakita kayong link na useful, you share it. For example, in relation to botany, ayan, just edit the tag so that those who would be interested in botany, they can just look at the tags. No? Ayan. And remarks or comments, pilot testing pa lang ito. This would link to the next sheet para tignan ano yung mga comment. Hindi po masyadong maganda yung interface or joke, wag ganon, wag malait. Okay, so for those who are here at meron kayong kunwari, may gusto kayong i-share din, you can actually place ganyan. And then posted by, ilalagay natin yan para if you have additional questions about the resource, you may ask them. Medyo hesitant pa ako to put the contact number, kasi syempre, data privacy. But in any case, at least there's a name there. At least hindi kayo zero, no? Sa kung in finding, no? Yung kung sino man yung nag-post, yung sa resource. Ayan, may naglalagay po, Canva, ganun. Although ano po ha, just a heads up din, kapag ka masyado maraming collaborators, I'm assuming ngayon madami kasi we have this synchronous meeting. Just a heads up po, kapag ka medyo marami, medyo maglalag. Expected na maglalag. Baka ma-delete nyo po yung nilalagay ng iba. Yan. So mga problems din na na-encounter ko with my class. Sir, may nilagay po ako sa may row 13. May dinilit po na ganito, tapos away. Mga ganon. And so for the purposes of this study, and for those who can, pwede kayong maglagay. Ayan, may naglalagay dito. Canva. Okay. And although ang nakikita ko dito sa aking spreadsheet ay anonymous axolotl. I think, ganun ata kapag yung email na nakalogin ay yung general or personal email. Ayan. And so this is just like a simple exercise. And for those who have access, you can always go back naman to this. No? Tingnan niyo ano yung mga nilagay siguro ng mga kasama ko. Ganun. Di ba? Affiliative, collaborative. Ayan, teamwork. Ayan. For creating impactful presentations. Okay, nice. Kahoot. Oh, yun. I'm not familiar with Kahoot. Maybe we can all look at this. Ayan. Nakakatuwa. Ang daming colors. Ang daming kong colors nakikita. Pero yun lang ha. Ulit. Oo, heads up. Baka makahanap kayo ng kaaway dito. Baka na-delete yung ninyo. Yan. Siguro para mas pagka may gusto kayong ilagay, tapos wala pang ibang naglalagay doon sa isang cell, go lang. But for the others, siguro later na lang, no? Pwede namang even after this session, tsaka kayo maglagay. Para maiwasan lang yun, baka magka-deletean po kayo ng entries. Yun din, one of the things that I'm really using heavily ay Google Spreadsheet. Una kasi yung shareability din, no? Tsaka yung real time. So when you collaborate, when you plan, when you edit, andun na siya. Even for example, yung sa yung sa scheduling namin dito sa SINSM, we're also using spreadsheet para kapag may changes sa schedule, I can change it. I can change it, and then my collaborators would see the changes real time. Ayan, Kahoot, quizzes, classical genetics simulator. Yes, I also encountered that. Also on the flies po yan, ayan. Tapos sasabihin na din niya kung ano yung mode of inheritance. So for example, kapag kayong sa may eye color ng Drosophila, it should be sex-linked. For some characteristics of Drosophila, Mendelian. So yung may mga ganun. Yeah, good. Okay, nice. Quizzes, gamified quizzes. That's nice. And for interactive presentation, ayan. Or lagyan natin dito yung lobster ni Ma'am EJ. Nakausap niyo na po ba si Ma'am Shahira? Tama ba? Oh, I'm listening, sabi ko sa inyo. Na representative. Okay, so kung makikita niyo baka may lag between or may discrepancy between your screen and yung naka-flash sa Zoom, no? Kasi syempre, internet din. Okay, so this is just a sample exercise and thank you very much. I really appreciate it. Na nilagay nyo, may mga nilagay kayo, may mga na-share kayo. Ayan. Ayan. Okay. So sige, I'll stop sharing this slide. You can continue. Mukhang nag-enjoy po tayo dun sa paglalagay ng paglalagay at pag-view ng mga available resources. Anyways, that's one way of collaborating online. Ang kagandahan po nito, parang binibenta ko naman yung Google. Ang kagandahan po nito, pwedeng asynchronous, nandiyan lang kahit na wala yung Zoom, pwede nyo siyang i-visit basta you have the link. No? But you have basta meron kayo ng link. And this is also a good time then na kasi I have educators from all over the Philippines. Yan. Na pwedeng mag-view at maglagay doon sa database na to. O yan, kahit spreadsheet lang po pala, kahit hindi na tayo although syempre may advantages yung mga softwares din. Pero for the sake of having a list of compiled resources, this will do. Okay. So ang dami ko nang okay. Check ko lang saglit yung sa chat. Okay. Thank you for sharing. Oh, thank you then for sharing kasi you're also sharing. Okay, so let's just go back to my presentation na hindi ko makita. Just a moment. Share. Okay. Mali. Tama ba? Ayan. Okay, so balik na tayo. Actually, this is second to the last na nung aking presentation. Affiliative approach. And we should champion collaboration. Ayan. Pero syempre din po, may mga resources din na exclusive for an institution. Yung mga resources tayo na exclusive for an institution, we should collaborate. Pero still, we would want to keep in mind na some institutions would have apprehensions, limitations in sharing their resources. Some resources are copyrighted and things like that. Yung kung ano lang po yung available. Ganun. Okay. Ayan. And as I mentioned, your databases doesn't have to be fancy. Okay. Ayan. So that's for the affiliative approach. And then last, ayan. Garage or home experiments. So yan. We also do this or binibigay din namin activity ito. Although there are some considerations or a lot of considerations when giving activities na gagawin sa bahay. So ano yung mga kasi meron. We can also look sa internet kung ano yung mga pwedeng nilang gawin. Yung mga pwedeng gamit ng mga household tawag doon, chemicals, ganon. Like for example, alcohol, ganun, or rubbing alcohol, or vinegar, mga ganon, salt, ganon, or ice, yung mga ganon. But anyways, as I mentioned, there should be some considerations. Number one, your experiment should need little to no supervision. Remember, gagawin nila ito sa parang parang art angel lang po. May pa-warning. Kailangan ng adult supervision. Ganun. Hindi, kasi students would be working with chemicals. Baka naman, syempre, high school and college, pero baka may mga kasama silang bata sa bahay, mapaglaruan yung mixture ng alcohol at saka nung plant extracts. Or baka naman meron yung mga immunocompromised or merong ibang condition sa bahay na sensitive pala, hindi dapat masyadong nakakalanghap ng maraming fumes, things like that. No? Basta yung kailangan ng little to no supervision, and of course, it shouldn't be dangerous. Next is, dapat yung experiments po natin ay hindi magre-require too much. Again, you should know the learner's context. Baka mamaya we require them to dissect chicken na in place of kasi isa po yan sa ma-share ko lang kasi I'm part of the zoology cluster. So sa dissection, 'di ba, we have ang tawag dito. Kapag comparative anatomy po, 'di ba, meron yung different representative species for major vertebrate taxa. So you have cat for the mammals, you have dati shark for chondrichthyes. Tama ba? Sorry, hindi ako on one. Ano nga ba yun? Tapos, meron din bird, di ba? Sa bird, usually, ang nakalagay kay Hyman, I think it's a pigeon. No? Pero, try chicken, baka mas available yon. However, there are limitations then. So I think my colleagues would agree, may mga problems with chicken. So, tingnan ninyo. And then, sorry, tawag dito. Lumayo na ako dun sa main point ko, which is baka we require them to buy some resources na mahirap, especially pandemic, may mga nawala, may mga estudyante po tayo na hirap, no? And then baka ipa-require pa natin sila to tawag dito, to buy certain materials na baka mahirap. So we should know the learner's context so that we also know ano yung pwede nating ibigay. Baka naman kung may poultry farm si student o di kahit magpa-dissect po tayo mga 5,000. So, 5 dosena. Basta, you get the point. I think you get the point. So, yeah. So, ano yung mga experiments? As I mentioned before, the internet is a good source, a rich source of home experiments. Isa dito, I think nakalagay ito dun sa shinare kong spreadsheet, home DNA extraction. This is interesting story. Sa FB ko lang po ito nakita. While browsing my newsfeed, lumabas, ang nerdy pala. So, ang nerd naman ng newsfeed ko. Well, anyways, at least it helped me. Lumabas sa newsfeed ko ito, itong home DNA extraction by just using banana and like ethyl alcohol. And one of my colleagues, Miss Po, she actually tried this. And yeah, it worked. It worked. Kailangan lang ay blender, dishwashing liquid. Yan. So yung mga ganon, mabilis lang gawin. Ano pa ba? Or yan, backyard surveys. Yan. So ito, sorry, hindi siya sa genetics, pero I'm also familiar with invertebrate zoology din kasi. So kunwari, backyard surveys, they just go to their doon sa, kasi baka syudad naman yung iba. They just go to their backyard and look at invertebrates, no, that they can see sa kanilang hometown. Tapos i-compile niyo yon sa class, di ba? So, para makita niyo sa iba't ibang sulok ng Pilipinas, makikita niyo yung mga species na nakikita nila sa kanilang locality. Baka may mga ma-observe kayong trend. Ay, yung mga taga this is for mga taga-syudad, more of ang nakikita nila mga periplaneta Americana. Cockroaches, gano'n. Things like that. And backyard survey, and home experiments such as home DNA extraction, Allium test. Allium test. This is also a simple test na pwede nilang gawin. Kaya lang, yun lang. Siyempre, may mga considerations. You need reagents, acetorsane, farmer solution para i-preserve siya if you can't process it agad-agad. Ano pa ba yun? Yeah, alcohol din, I think. Pero baka naman pwedeng ang gawin nyo lang for the Allium test ay, hindi na siya Allium test per se, pero baka pwedeng yung macroscopic na lang, yung macroscopic parameters ang measure ninyo. For example, you have different water samples and then just measure the root length. Yun, doable yun sa bahay. And then subject it to chi-square analysis or other statistical tests. Applicable statistical tests. So yun, may mga ganong considerations. Okay, so I think that's the last slide, no? Last slide for the lecture portion. Yan. And I hope marami kayong natutunan dito sa lecture. Okay, so after this, Actually, nag-overtime nga po ako, pasensya na. After this, we have the lab demonstration. Not really a lab demo, more of tour. Ganun. I'll just try to show you ano yung itsura doon sa different links, no? Links na ginawa natin. So actually, short lang yon. Oo, short lang yun. Kasi naman, yung the nitty-gritty of the virtual labs, mas matututunan nyo siya if you play with it. Na kapag kayo mismo yung nagpipipindot doon. No? Kasi kayo nagmitir-mitir ijay kung na Ilocano. Okay, so that ends the lecture portion. May we have a short break muna? No? Let's just have a short break muna, bathroom break. Yan. So, yeah, maybe 10 minutes, 15 minutes. Okay. So, yan lang. See you after 15. Okay, and we're back online. And I hope nakapag-merienda na po kayo at nakapag-refresh. So we will have some adjustments in the lab demo portion. Mostly I'll just breeze through, no? I-scan lang natin yung ibang na-mention na labs, virtual labs, simulations. As I mentioned, you can also play with it in your own time. So, sige. Let's just start the next presentation. Ayan. Hi. Okay. Ayan. So, for the remaining portion of my talk, I'll just mention some resources that you may use in your genetics activity. Ayan. So, ito lang, this is just a brief background on some concepts related to one of the activities na pwede natin gawin, which is the Allium test. Ayan. So, Allium test is actually one of the assays that can be used to test mutagenicity. Okay? Okay. And so just to provide a background, pag sinabi natin mutagens, yan, this will be those substances that would cause mutations in DNA of an organism. Mutations, changes. Yan. So from wild type, no, meron siyang tawag dito, iba't ibang uri, no, ng changes na pwedeng ganap. Yan, mutations are naturally occurring in nature. Ayan, pag sinabi natin mutation, it's not necessarily bad. It's not necessarily bad. It's actually naturally occurring in one of the sources of variation. Okay. Bakit ba natin kailangan ng variation? Actually. Sige nga. Short lang po. Tignan natin, sinong first honor? Bakit po natin kailangan ng variation? Comment below. Check natin kung bumalik pa po ba kayo o may mga naghuhugas pa po ng pinggan. Ganun. O yan. Sir Kenneth 3 increases fitness in the environment. Yes. Adaptive characters. Yes. Biodiversity. Yes. Tama. Parang di ba kapag may variation, you have richer genetic arsenal. Ganun. Para imagine kapag walang variation, iisa lang yung genetic component ng lahat and then there's this catastrophic event, wipe out yung buong population. And as a parang safety, life would have these designs, no, napaka ganda. With variation, yung mga ganong safety nets. Anyways, yan. So evolution, sige. For improved variety or breed, yeah. Increased survival, yes. So that's the better side, no, of mutation. However, of course, there's also the bad side. No, there would be these mutations that would cause diseases. No, would have debilitating effects on the organism. So that's why we also routinely check, no, for mutagens. Okay. Yan. So mutagens would affect DNA in various ways. Yan, change DNA sequences, cause chromosome instability, and cause duplication or deletion of whole chromosomes. And so how do we assess mutagens? And we have several assays. And so these assays, I think Ma'am EJ would be better, would be the person to better explain, no, some of these assays. And so you have Ames test, micronucleus test, Allium test, Comet assay, and of course, I'll be focusing on the Allium test, ang walang kamatayang Allium test genetics, at bote ng Yakult, or kung ano mang ginagamit po ninyong bottles. Okay, so Allium test, just a brief background. It makes use of a plant system. No? So the model organism here is the onion, no? Allium cepa. For, take note ha, yung pag-write po ng scientific name. And for assessing environmental toxicity. And so dito din na makikita yung crossroads, genetics, ecology. Of course, since mutations would also have involvement with carcinogenesis in other diseases, it's also related to also have obvious applications in the medical field. Okay, so it's developed by Gerd Fiskesjo in 1985. Actually, nakita ko from the net, just search it. It's yung paper niya ay open access naman. And so you can actually review that document. And so utilizes through tips of onions where mitosis can be observed. Yeah. Okay, so these are the things that na tinitingnan sa Allium test, no? Yan. So mitotic activity, more or less you have this, you assess the mitotic activity. And somehow by comparing it to a control, we will see if the water sample na ginagamit ay may mutagenic effect. But of course, syempre it's too general kapag Allium test lang yung ginamit. So usually Allium test is used in conjunction with other mutagenicity assays. But for teaching, maganda siyang gamitin. Actually, even for research, better pa rin lang na may kasama siyang ibang assays na hindi lang Allium test yung ating basis. Okay, so ito, we have the test organisms here. Yan. And then these are just some of the parameters that we're checking. In broad strokes, you have two macroscopic and microscopic parameters. For the macroscopic parameters, root length, root form, and even shoot length. Pag lumabas na yung sprout niya, ganun, laso na. Gamitin din. Anyways, yeah. Tapos, for the microscopic parameters, ito yung mas fino-focus, hina-highlight. Yeah, we check for the mitotic index to somehow assess the mitotic activity no ating model organism. And then we also look for chromosomal aberrations kung present. So some chromosomal aberrations, you have these, C mitosis, vagrant, sticky chromosomes, bridges, and fragments. Okay, so let me just clean my screen. Okay, so you're familiar with this. Pinakita ko na to kanina dun sa my lecture portion. And as I've mentioned before, as an activity for the students, you can show them a picture and then maybe ask certain questions. For example, here, parang magbigay ka ng hypothetical situation. This is a slide of onion root tip. Ayan. And then you can ask them to determine the mitotic index. Ayan. So like yes, manually they need to count. So pag sinabi natin mitotic index, it's just the proportion of the mitotically active cells. Tama, it's the proportion of the mitotically active cells. So paano na natin yun makukuha? Mathematically, you just need to, of course, total muna yung cells that are undergoing mitosis over the total number of cells in that field, no? In this field of vision. Okay. Yeah. Okay, nakuha. Yan. Tapos, at saka ito, medyo dito din papasok yung training kasi there would be, it's for some students, it would be quite challenging kung paano nila malalaman yung difference ng prophase saka interface. Yung dalawa doon, no? Medyo yun doon sila nalilito. They would label a cell undergoing interface as prophase. Kasi nga naman kapag ka-prophase parang wala pang obvious na action na nangyayari doon sa ating chromosomes. However, yan, ang isang magse-separate would be yan, cells with visible chromosomes would be those that are actively participating in the mitotic process, no? Mitotic process, tama. Kasi karyokinetic process, it's just the nucleus, no? Parang gumagalaw yung, ang tawag dito, nagse-separate yung genetic component. So yeah, cells with visible chromosomes would be those that are undergoing mitosis. Cells without visible chromosomes, those would be the cells in the interphase stage. So remember, kapag mitosis na, dapat condensed na yung iyong genetic material, di ba? Dapat condensed na sila and packed into these units. So units called chromosomes. Okay. So in this example, ayan, what's the mitotic index? Sige. Ayan. Ayan. So, ganyan. Ayan. Thank you pala, Ma'am EJ, for providing this resource, this picture. Ayan. So, makikita natin dito, we can see those cells that are actively dividing, those that are in mitosis, mitotic stage. Yan. So, ito, what specific stage of mitosis? Ayan, minsan may dance step pa yan eh. That be your? Ay, talaga naman po. Ang gagaling. Anaphase. Yan. Personally, ito yung isa sa mga na-excite ako pag nakikita ko siya sa slides. Anaphase. And for some, ito na medyo blurry na. Hindi mo na masyadong makita. No? Anyways, for mitotic, ito. Number 5. The cell in number 5 is undergoing what stage? Metaphase. Diba? Isa rin to sa mga na-excite ako. Yan, so pwedeng yung mga ganito and then obviously you can get the mitotic index, but also you can have other questions. For example, what based on this figure, aling specific stage ng mitosis yung pinaka-konti? So they would have the data. And then ask them why. Ganon. So they need to research. Bakit nga kaya? Yan. So most probably, yung mga konti lang would be yung mga nasa, okay lang, alam na nila yung sagot kung sakali. No? Anyways, anaphase, yan. Tsaka metaphase, kasi medyo transition. Na in terms of yung bilis, kung kailan siya mangyayari, mas mabilis mangyari itong dalawang to, especially yung sa metaphase and anaphase. Tama, metaphase and anaphase. Anyways, yan. So we have this counts and then we can get the mitotic index. Usually, minumultiply ba siya by 100. So in this case, with this slide, the mitotic index is 26.7%. Okay. Yan. So, medyo napakita ko na rin ito kanina doon sa my lecture portion. No? So, if this is one of the activities na pinapagawa namin with students, we provide them with slides of our onion root tips. And then, if this is field of view 1, 2, of hindi ko nalagay yung label, of same onion root tip so that they can get the mitotic index of that onion root tip. Remember, usually we score 500 cells. Pero minsan, depende na din. Minsan binabawasan namin. But 500 cells, to score 500 cells would be a good number na. Okay, so meron din, may instances, mahirap, we can't find good pictures for that. Ang isang pwede ninyong gawin is to look for researches using Allium test and yung dive straight to the results nung journal and then look, especially maganda kapag may table, makikita natin ganyan. You can just provide them with the table and then again, through tactical use of questions, meron kang ma-achieve na learning objectives. For example, this is from Nefik et al. Sa kanilang research. So, ganun din pala kapag gagamit po tayo ng journals, we cite them. Yan. So, they would have this data. Parang ang daunting naman ng ganitong data, pero okay lang yan. May mga learning curves lang, pero kayang-kaya yan. And so for this, makikita natin, of course, they should have a control. And then, the context of this is that they are trying to check the mutagenic or mitodepressive mitodepressive impacts of alprazolam, which is a medicine, which is a medicine used for, I think, to treat or manage panic attacks and anxiety disorders. And so anyways, and so you have different concentrations of that chemical, and then they try to look at the effect on the mitotic index. So syempre dito, you need to have the control. Kasi nagre-range po ang mitotic index ng ating allium cepa, nagre-range po. Kaya importanteng meron tayong control, yung wala kang ibang dadinagdag, just water, distilled water. So makikita natin there's a trend. Yung mga ganyan, no? Ano yung makikita nilang, ask the students, ano yung trend na makikita nila with increasing concentration of alprazolam, what's the effect on the mitotic index? So somehow, ano yung effect niya sa mitotic activity ng ating onion root tip? And makikita natin, there's a trend. While when you increase the alprazolam, it also inhibits the mitotic activity. So merong mitodepressive effect si alprazolam based on this result of Kinesthetic et al. Yan. Yeah. Okay. So again, these are some of the things that we test for Allium test. We have macroscopic parameters and microscopic parameters. Yun din. I showed this again kasi I just want to emphasize the point that if testing for microscopic parameters is really not doable with your current situation, maybe one of the activities that you can do is still conduct the Allium test and then just measure the macroscopic parameters. For the benefit of everyone na rin pala, kapag ka-Allium test, basically you will test water samples or chemicals, and then you will use the onion as your test organism. You would place them into these water samples, and then kasi kapag ka nabasa yung ilalim ng Allium cepa mo, mag-grow yung roots. Mag-grow yung roots. Ngayon, roughly, makikita mo din if your test chemical would have mitodepressive or mitogenic effect. Parang, syempre kapag may mitodepressive effect, makikita mo yun sa roots. Kahit yung macroscopically lang, makikita mo baka hindi masyadong nag-grow. Pagka wala masyadong effect, dapat normal lang yan. Medyo hahaba yung roots. So yun, pwede mong ma-compare. Parang baka pwedeng ma-observe ng mga bata, pagka ang ginamit ko po palang water ay from like canal, gano'n. Ano po ang effect, gano'n. And then they can just research on that, ano yung pwedeng effect sa cells, sa root cells, gano'n. Sa lab, we instruct our students to have different samples. Ang instruction nga namin, mas madumi, mas maganda. Baka mas may makita kayong differences. Baka may makita kayong differences. Ako ba yun? Bakit may streak dito ng blue? Anyways, tapos we also use EDTA, parang dun sa mitodepressive na element, na subject. Ano? Na chemical. Sorry. Okay, anyways. Ayun. So next, ayan, dun sa microscopic examination, liban dun sa pag-assess ng mitotic index, we can also look for aberrations. So again, thank you, Ma'am EJ, for providing this slide, this image. So makikita natin dito, you have yung first column would be the for reference, yung normal na itsura dapat ng ating chromosomes. And then you have these aberrations. Yeah. Tapos dito kung ano yung tawag sa aberration. And so for Allium test, positive result is a decrease in mitotic index. So ibig sabihin, if your test chemical decreases the mitotic index, that would mean baka may mutagenic or cytotoxic effect yung chemical mo. Yan. And in addition to that, you also isa ring positive result is if you have observed a chromosomal aberration such as these guys. Okay. Okay. Yan, so just some details on Allium test. Yan, useful assay for mutagenic potential assessment. Yan, cheap and very easy to perform with basic lab equipment, microscope. Yan. Yan. Of course, important that the researcher is familiar with basic cell biology. So for example, the morphology of cells undergoing mitosis. Kaya kailangan i-train din natin yung eyes natin. Some limitations. Yan. Although yan, Allium test should be part of a wider set of toxicological tests. So medyo mas strict yung rules kapag ka-research, for research. But for a class activity, kahit hindi masyado, we can modify. We can modify. And so just to mention some okay, so some researches that would utilize tawag dito, Allium test. Ayan. Ay, sorry. Anyways, I will provide naman the slides po. Okay, so next, sorry, medyo bibilisan ko na po ha. So next is the fly lab. Isa po sa nakita namin pwedeng gamitin for our experiments in lieu or in replacement for the mating ng ating flies. We have the fly lab. So very quickly lang, let's just try to look at the interface. Fly lab. Yan, so ganito po generally yung itsura niyo. You can design your flies for mating. Makikita na po ano. Yan, you can design your flies for mating. Yan, so you have different parameters here. Anong phenotype ang gusto ninyong ibigay dun sa inyong parent organism. So for example, for the purposes of this demo, quick demo, for example, eye color. Yan. So eye color, yung female ko gusto ko ay meron siyang siguro wild type or hindi. Sige, wild type. So I'll select that for mating. And then for my male, I will select maybe also for eye color white. Ayan. Then select that for mating and then you have your flies. We try to mate the flies. Okay, so ang lalabas, we have this. Ito na yung offspring, napakabilis lang. You have your offspring, yan, females, 519 are females na wild type, na red-eyed. And then for the males, you have 490, no, na also red-eyed. Yan, so you can analyze this further. Yan, just click on the tab on the Analyze tab. So it will show you a summary table of the cross, no, of the simulated cross. And then you can also, may mga options lang ito. You can, do you want to ignore the sex of the flies or not, depending on ano yung gusto mong i-discuss with your students. Yan. Or you can also include test of hypothesis. Ayan, you can also subject it to chi-square test na agad. Yan. So based on kapag sex-linked yung tinitignan natin na mode of transmission, of course, you would have your hypothesis. Your hypothesis would be the ratio of the offspring should be like this. Tama? And then you would put that in the hypothesis. Yun yung magiging expected na percentage. So with this cross, dapat ang expectation ay 50% ay wild type female. With the cross na ginawa natin, 50% ay female, while red-eyed female, 50% din ang red-eyed male. Yun yung based on the rules, no? Or yung kapag ginawa mo yung cross, ito dapat yung ratio nila. And then i-test nito ngayon with the observed number. And then let's test the hypothesis. Ayan, lalabas yung chi-square statistic, yung calculated, no? Yung calculated na, ang tawag to? Calculated na value calculated chi-square statistic. And then you need to compare that with a tabulated chi-square statistic. Okay, so in this case, we're just interested with this. Degrees of freedom, syempre, number of your phenotypic classes minus 1. Diba? Tatandaan natin to kasi ito yung gagamitin natin when we will refer back to the chi-square table. Okay, anyway, so this is your calculated chi-squared statistic. Let's just compare it with that of the table. So babalik lang ako dun sa aking presentation. Ayan, so ito yung ginawa nating cross actually. Hindi, reviewin na lang po natin to yung concept. Yan. But anyways, ito yung ginawa nating cross. Tama? Pure breeding red-eyed female with white-eyed male. Yan. So ito yung lalabas dapat. No? Ito po dapat yung lalabas. And then tignan natin if ano yung lalabas 0.8 something. No? And then we will compare that with the tabulated value. Let's just say ano po ang gusto nyong critical value. Ito rin minsan depende po sa inyo. Pero ang sa literatures mostly nasa 0.05, no? And then degrees of freedom 1. So ganyan. So compare yung calculated with the tabulated. Calculated is 0.8 something, and then the tabulated is 3.841. So your calculated chi-square statistic is less than, diba? 8.81 is less than 3.84. Therefore, accept your null. Ibig sabihin, in your null hypothesis would be this. Observed values fit the expected values for an X-linked mode of inheritance. Actually, you can change the phrasing of your null hypothesis. No? You can change the phrasing. Basta ang gusto lang nating sabihin kapag null hypothesis, walang significant difference between the values that you're comparing. Okay? Okay. So yun na for the fly lab. Actually, that's F1 generation pa lang. Tapos what if we will have the F2? Posible din naman po yun doon sa fly lab. Like for example, ito, we will return to the cross. We just select this. We just select this and this for the mating. May lalabas din pong results. Yan. So ngayon with these parental generation from yung F1 natin na pinag-mate natin. Now we will have 3 phenotypic classes, including the sex. Okay, so you have the table, and then you can also include the test of the hypothesis, etc. Okay po? Yan po. Next, very quickly na lang po yung iba. Yan, PopGen Fish Pond, simulation din po ito. Mali. Ayan, so for the basics of the PopGen, may nakalagay dito, yan. Background, yan. Ano yung Ayan, so anyways, ito po. These are the forces, no, of evolution that can affect with your Hardy-Weinberg equilibrium. And then, yan, you can run experiments. Pero minsan naglalag. It's not as beautiful as I've mentioned. It's not as detailed as those of paid virtual labs, but you know, it will still, it's useful. It's a useful tool. And so you can just tweak, no, some of the parameters here, and then titignan yung observation dun sa pond. Yan. Pero sa akin personally, mas gusto kong tignan yung dito, yung data, anong mangyari. So for example, what if I reduce the population na pwede lang, yung population nitong pond na to 25. Effectively, if you reduce the carrying capacity, you will reduce also the total number of population that can be supported by this pond. Okay, so basically, gusto nang sabihin, lumiit yung population dito. Tignan natin ano yung magiging effect. Wait lang, reset natin. Ano yung magiging effect, pabilisin natin. Kunwari, yan, 16. Okay, let's go to the data. Siyempre, ang initial population natin ay 200, tama? So, it's expected na after several generations, we reduce the carrying capacity, babagsak talaga yan. Pero ito po ang mas importante nating tignan, ano yung trend. Bumubulusok ang likot nung pag-change sa allele frequency and genotypic frequency. And that is because we have, di ba, kung babalikan natin concepts, mababa yung population. If we will try to review one of the assumptions sa Hardy-Weinberg equilibrium, the population should be infinitely large. Para negligible na yung effect ng genetic drift. Ayan. So yung mga ganon, we can review all of those things with these simulations. Namatay na po ata yung aking fishes. Ayan, so you can tweak all of these, these things. And so migration, mutation, etc. Okay, so siguro last na lang po yung isa. Gusto ko lang talagang i-highlight kasi personal favorite ko na po yung learn genetics. Okay, next, share screen, learn genetics. Yan. So, pwede, you have the link naman na po to this. It looks like this. Yan. Learn genetics. Ang isang pinaka-natutuwa po ako dito, ito, make a karyotype. You have interactive resources here. Yan lang minsan kailangan mag-load. Ayan, pwedeng ipa-activity natin or pwedeng tayo din if you want to review. Yan, so we have here a karyotype. We just need to gusto ko ng hint. Medyo nahihirapan ako minsan. Ayan, ito ba yung kapartner niya? Ayan, so things like this. You can do things like this in this site. And then you can just review. I think meron na ring mga links to the concepts na relevant to this activity. Okay, so yun lang po. I think that ends my the lab demo portion of my presentation. Again, thank you very much. Thank you very much for staying with us, even on a Saturday. Maraming salamat po.
Okay, maraming salamat po Sir Jeno sa inyong very insightful and informative na presentation. Okay, so ayan, hitik na naman po tayo sa dunong na naibahagi po ng ating resource speaker, no? Lalong-lalo na po sa pag-handle ng genetics in an online setup. All right, thank you very much Sir Jeno for sharing experiences and knowledge in teaching genetics in an online setup. So maybe we just try to have a recap lang po muna, no, of what transpired during his talk while we are maybe developing our questions for the resource speaker, of which I invite everyone to please post your questions po sa ating chat box so that we can, mayroon po tayong small talastasan or open forum para po sa discussion natin for today. So just a recap, no? Sir Jeno highlighted the new normal approach in an online learning with focus on leveling off with our objectives and expectations to our students. As of course, our experience during this pandemic is a case-to-case scenario. He also gave some tips on how to effectively handle online class. So talagang naisabak po tayo, no, bilang mga teacher, lalong-lalo na po yung mga kaguruan natin dito na nasa basic education curriculum. So though we have these challenges po, talagang it only proves na we really have to be resourceful and creative, no, in order to deliver and meet the minimum standards ng ating mga subject, for the sake also of at least not compromising po, no, yung mga lessons at mga dapat malaman ng ating mga estudyante. Okay, so may mga accessible link din po tayo provided through Google Meet that everyone can access, at maraming salamat po sa inyong aktibong partisipasyon. May bahagi din po sa atin ni Sir Jeno ang importance ng Allium test for genotoxicity evaluation. So ito po, sobrang ginagamit po namin to sa UP Baguio, especially po sa undergrad genetics course namin. And very useful and insightful po yung methodology na ito just by, using onion, no? Siguro, and then we can place it on top of a Yakult container, then we can already assess or ipakita sa ating mga students po, no, yung genotoxicity potential ng isang source or maybe a water or anything that we would like to assess. Siguro yung pinaka-challenge po talaga, no, sa pag-conduct po ng Allium test natin is really the technique in the preparation of the slides, no, because of course that the cell should properly be mounted and at the same time fixated para po maayos yung pag-review natin under the microscope. Ganon din po na ibahagi ni Sir Jen ang ating fly lab. Ayan. So siguro magandang avenue din po yun para may turo natin sa kanila ang basic na genetics basic genetics theory, especially sa breeding or concepts of carrying capacity, something like that. Ayun po. So okay, so let's try to check for some questions that our participants have posted under the chat button. Ayan, so ayun. Maraming salamat daw po, Sir Jerry. Nag-accept po siya ng baka may gusto pong magbigay ng tulong via GCash. Joke lang. Ayun. Okay, or maybe sa YouTube natin. Ayan, may top commenter for today po tayo. Sorry po, para po ako nag-a-announce na pabingo. Okay. Okay. Ayan. Shout out po kay Maria Feliciana Benita Elareta. Yeah. Okay. Okay, a question from Professor Dave Michel Lantoria. Sir, may I ask po if this application may be pertaining to Fly Lab mentioned are free and does not require any subscription pay?
Yes po. Ang kagandahan ni Fly Lab is that it doesn't need any subscription. It's an open educational resource. So you just need to go to the site and then, you just do your simulations.
Okay, thank you for that, Sir Geno. So, wow, napakaganda naman pala ng application na ito at it's royalty free, no? At magagamit po ng basta meron po tayong internet connection. Okay. All right. So you have mentioned a question from Sir John Raymond Torres. So you have mentioned about understanding our learners' context. How do we go about their schemes or even ninjutsus? Oh. What I mean is, how do we objectively assess if some of the things they are presenting are already just excuses to avoid work or assignments? Then, okay.
Yeah, thank you, sir. Opo. I think I can also relate with this concern. Oo. Kasi first and foremost, we can't be sure kung totoo ba yung sinasabi ng mga estudyante. But I think one thing na pwedeng gawin is ako sa akin in my classes, I do class surveys to better understand their context. And may part doon na I just need to trust them. Kasi as educators, I would err on the side na talagang may pinagdadaanan yung bata. Kaysa I would think na he or she is just making excuses. Kasi kung titignan kung ano yung magiging net effect, what if the student is really in need of consideration and ako sa akin na feel ko siya na meron siyang mukhang hindi yan totoo. Medyo damaging yung magiging effect. So part of it is really to trust them. Pero I think for more objective or parang mas informed tayo, usually I conduct surveys or minsan I ask the friends or I ask some of their peers. Kaya malaking tulong din po na you divide them into groups. Sila yung nagkakamustahan din. More than anyone, they would know the real situation of their peers. Yeah.
Tama po. Kasi may mga instances po talaga na though lagi natin sinasabi sa mga estudyante natin na you can always approach me if you have a problem, something like that. Most of the time, they are more open to sharing their thoughts, their feelings sa kanilang mga friends, peers, at mga group members. So, ayun po. Tama po yun, Sir John. We have another...
So just to add, and kawalan kasi nila rin yun. It would be their loss kapag they try to dodge and go out of their way to dodge a learning activity.
All right. Thank you, sir. Additional question, very interesting from Ma'am Josephine Joy Tolentino. So thank you, Sir Jenna, for that very insightful talk. Just want to ask, do you have any suggested alternative stain to be used in Allium test aside from the aceto or sein? Our campus don't usually produce aceto or sein, and my students have a difficulty in looking for the stain. Thank you so much in advance. Yeah.
Thank you, Ma'am Josephine, for this question. As of now, wala po akong mai-suggest na ibang stain for aceto or sein kasi for the longest time we're using aceto or sein. But I think some of my colleagues tried to use red cabbage extract, I think, but I just need to follow up ano yung result nung experiment na yon. But I'll just give you an update on that maybe after this. Yon, I can't give an alternative, but I can only share na trinay ata nung sa Bio 100, I think natin, Mams, na gumamit ng red cabbage stain. Makikibalita lang ako kung ano yung nangyari.
Okay. All right. Another question from our YouTube participant, Ma'am Eloreta. So, for this simulation modality pertaining to Fly Lab, how much time should be allotted po siguro, sir?
How much time will be allotted for the students? Yung kapag sila po yung gagawa. If ang assumption ay for the students to do it, depende din po kung ano yung tinatanong ninyo. Kasi depende kung anong cross yung ipapagawa ninyo sa students. Kasi there will be some crosses that would medyo parang mabilis lang, parang laging 50% lang lagi nung dalawang phenotypic classes ang lalabas. So with that, siguro mga 10 minutes, ganon. So, ito just on the top of my head pero again, depende ulit din sa sitwasyon ng mga bata. So, sa akin 10 minutes kasi siguro I have a background on that. Pero kung sa estudyante na zero knowledge pa lang sa X-linked mode of transmission, maybe more time, mga 30 minutes. Or depende kung ilan din yung crosses po na ipapagawa natin. Pagka mga nasa F3 na po yung papagawa natin, medyo mas komplikado yon. So maybe we will give them more time.
All right. Okay, thank you, sir. Another question from a YouTube participant, Ma'am Pula. For the Allium SEPA assay po, sir, is there any specific time for us to visibly see the real-time mitotic events?
Ay, sorry. Pwedeng paano? I didn't catch that. Sorry. Medyo nawala yung connection ko.
Sir, parang gaano ho kaya sila katagal mag-aantay, sir, to visibly see the mitotic events once na pinerform po nila yung assay?
After the staining. Pagka na-stain, tas gaano katagal? Let me just check with well, actually, we have a protocol, but kung di ako nagkakamali, 15 minutes for the staining, and then you can view it under the slide. Kaya lang, we also have the, just a note, there's also a danger of overstaining. Baka wala na po tayong makita, lahat na po ay pula. Yan. So with that then, actually, ganito po. It's interesting. Ang ginagawa namin is, we have, tawag dito, yun, ang original is 15 minutes. Yun nasa protocol. However, after that first run, we will examine lahat kami doon sa klase, and then titignan namin yung quality. And then if it's overstained, then for the next, babawasan po namin into 10. And then kung overstained pa rin, babawasan namin into 8. But usually 10-15 minutes is okay. Minsan kasi depende rin dun sa pagkaka-prepare ng aceto or sein. That's why we need to tweak these things. But normally, yan, as a starting point, 15 minutes of staining. And then adjust na lang. Kapag overstained, bawasan. Pag understained, usually, kapag ka-understained, it's not a matter of time . Mas ang nag-matter minsan ay yung pag dapat freshly prepared ang aceto or saline stain.
Okay po. Other question from a YouTube participant. What about collaborative activity po? Kasi they to be connected. Ay, wait lang. Paano po ito? Maybe Ma'am Princess is asking about tips po on how we can conduct collaborative activities in a way that all our students will be participating po, no? So parang i-avoid natin yung free rider, something like that. Sir Jenna, nakamit tayo.
Ayan. Yan. So yeah, for collaborative activity, usually ganun, we instruct them to have their smaller groups, parang peer groups. And then sa akin, ang ginagawa ko, and my colleagues din, you assign one person per group na point person na siya yung may task na kamustahin yung mga peers niya. Kasi ganito din, isa sa mga pwedeng gawin is if you give them task, if you give them accountability, they will actually work. Yun yung isa. So if you identify one person, more often than not, he or she will do the job. Follow up niya yung mga kasama niya para mas collaborate, mag-collaborate sila. Yeah. Parang you set up subunits of the class, and then may mga may mga leaders doon sa mga subunits na yan. If you divide them into those smaller groups, mas madaling mag-reach out, mas madaling i-engage yung students.
Yeah, okay, tama po. Thank you, sir. Siguro sa may tanong lang ako, or maybe clarification for everyone na in the event po na hindi po available ang Allium for the Allium test. Do we have, or can we propose other alternatives that we can use for this endeavor?
Admittedly, I haven't tried any other model organisms. Kasi Allium would be, yun na yung readily available, tsaka mabilis relatively yung pag-grow. But maybe, yeah, na in relation to genotoxicity kasi baka, I'm just on the top of my head again, maybe you can use mung beans kasi mabilis din siyang mag-grow. However, yung growth nun ay hindi masyadong dependent on the quality ng water na ginagamit mo, basta may moisture and yung sa light yung titignan. So yun, the short answer is wala. Wala pa akong masabi na effective na alternative with onion.
Alright, okay, sir. Siguro tip lang din po, no, for our other participants here na when you do the Allium test, kailangan din po natin mag-prepare no, ng positive tsaka negative na control. Wag po natin kakalimutan. Ayun, sir. Sir Jenna, you want to add something from that?
Apo, tama po yun. Just to add to that point, hindi pwedeng wala tayong control kasi that would be our reference point. As I've mentioned before, there are parang sa mga literature, merong naka-indicate na ano yung expected na mitotic index for a control, but you can't use that for your own setup. Kasi iba-iba yan. Like for example, yung onion bulbs na ginamit, baka hindi magkakabatch, baka mas mabagal yung pag-grow ng roots na ginamit dun sa binasa mong article, ganun. So yeah, importante meron kang reference point. You have control, positive, negative controls.
Thank you, sir. Are there more questions po from our participants? Here, another question from Sir John Raymond Torres. May we know what is the best variety of onion to be used in the Allium test based on your experience in the laboratory? And follow-up question po doon is, in the assessment of macroscopic features of onion roots, if we are aiming for toxicity, is it really needed to have a pre-rooting stage first before subjecting them to test solution? Will it be more appropriate and fair if we just go directly to exposing the test organism in the test solutions po?
Okay. Sige po, thank you po. Thank you sa question. Yan, so best variety based on experience yung pong, hindi ko alam kung anong tawag sa variety na yun, pero yung red na hindi sobrang laki. Hindi ko alam kung anong name pa, sorry ako. Pero we also tried yung puti, tsaka yung mga malalaking onion na red. Most, hindi sila masyadong, kasi meron yung mga malalaking onion na red, hindi sila masyadong nagpo-perform sa experiments. Yung mga average size na red. Oo. Sorry, hindi ko alam yung tawag doon. Pero yun po. Yung nabibili sa Garita. Sa stores.
Sir, kwan pa, yung follow-up question ni Sir John Raymond is, do we need to have them pre-rooted or we can just expose them directly po to the medium of the test?
Better po sana if may pre-rooted sana, so that you're sure that before you subject them to allium test is viable yung cells, viable yung onion. Para sure ka na actively ay mitotically may potential pa yung bulb. So para sure ka. Kasi baka kunwari, hindi natin siya sinubject to pre-rooting stage and then nilagay mo siya sa test chemicals and then walang lumabas na root. Hindi tayo sure na dahil cytotoxic yung ginamit mo, baka kasi hindi viable pala yung onion na ginamit. So better sana if may pre-exposure muna before you put it. So that's one of the considerations. Kasi meron din, well, totoo din naman na meron na yung mga kahit na kapag ka diniretso mo siya, wala na talagang mag-grow. Kaya lang yun lang, hindi tayo sure if that can be attributed to that test solution or the viability of the onion bulb.
Alright. Ayun, tama nga Sir Jeron, no? Sometimes we even try to scrape muna the bottom portion of the allium so that it will expose the actively dividing cells para ma-trigger yung generation po ng root. Kasi once na nagkalos na yung the bottom portion of the allium, sometimes hindi nag-grow yung roots. So tama po, pre-exposure is very, very important to ensure na talagang may ugat. Na may ugat po tutubo doon. Yeah.
That's right. Dapat tatanggalin po yung brown portion but not too much. Again, yun din subject to dapat ang tawag doon, matrain kung gaano ka deep yung kailangan tanggalin. Baka po tinanggalan natin pati yung actively dividing, hindi po tayo magluluto. Hindi po siya panggisa. Tatanggalin lang po natin yung dead cells para mas ma-encourage yung growth ng roots.
Ayun. Yeah. Right. Tama po. Okay. So may nagpa-pop up dito sir na question about the mitosis dance steps. Ayan. Baka pwede daw sir pa-sample dahil patapos na lang din naman tayo.
Okay. Actually, in relation to that, in my classes, sometimes I do awkward things like this. So, actually, we're incorporating kinesthetic element kasi may dance. Pero sige, ha. Yeah, I do this. You have your pro face. And then, sunod, meta face, ana face, yellow face. So you have that dance step. Tinuturo ko to. Especially pwede kapag online kasi hand tatting, hindi mo kailangang tumayo. Di ba? Parang, ganun lang. Tapos i-attitudean mo na lang na ganun. Nasa YouTube nga pala to, no? Okay.
Sisikat ka na, sir.
You can remember, actually nakakatulong yan. You can remember things better if you have these mnemonics or you associate, if you engage more senses. So hindi lang visual, hindi lang auditory, you're also incorporating kinesthetics, movement. Yeah.
It's a form of mnemonic, no? So especially sa mga bata sa high school. Kasi nabobore sila, ano ba itong pro-face, meta-face, ana-face. So lalagyan natin ng konting angas. Yan.
Naging dance step.
Maalala po nila.
Just to share din, sa lab exam namin may sumasayaw.
Paano daw pag aberrated na yung chromosome?
Ano, no?
Paano daw sa pag aberrated na yung chromosome? Paano yung sayaw niya?
Wala, mangisay na lang na gano'n.
May kulang sa isang paa. Yung chromosome. All right. Okay. Are there more questions entering in the YouTube platform?
Chromosome bridge.
Okay. Let's have to browse other more chats. If there are anything na hindi ko pa nabasa. Okay. So while waiting,
may we acknowledge po pala the presence of our VCAA and VCAA Rosemary Gutierrez. Hello po.
ayun. Hi, ma'am. Morning po.
Good morning.
Ayun.
And
Huwag po
good
tayong
morning
mahiya.
to the participants.
Oh, here, another interesting question, Sir Jeno, no? Are there any environmental factors for like humidity, temperature, etc. maybe participating in the overall final output or yung effect ng Allium test po natin, according to Sir John Alex?
Yeah, definitely merong effects. Pero ang hirap i-single out din. That's why we will have the control. We will make sure that the control would have same environmental conditions as the other test bulbs. Para makita lang, di ba, i-minimize natin ano lang yung i-change nating variable per setup. So yeah, definitely to answer, definitely there's an effect, but for us to not think about those environmental parameters like temperature and humidity, we have the control. We have the control setup.
Okay. All right, so I think that's already the last question that we have for both the YouTube live and our open Zoom chat. So, ayun, marami pong salamat sa pagpa-participate sa ating genetics module with our resource speaker, Sir January Pagaduan. And though unfortunately, we'd still want to continue with this activity, but everything needs to, we have to cap off, the 3-day seminar workshop that we had for this 26th SINSM. Okay, so to formally close, but of course there will still be an awarding ceremony later on after the closing remarks for our speakers that will be presented online po yung ating mga certificates. May I please introduce to you our very own biology professor, a specialist microbiologist, and our current Vice Chancellor for Academic Affairs, our former Dean in the College of Science, Dr. Rosemary Gutierrez, ma'am, for our closing remarks and message to our participants. Thank you, ma'am.
Thank you very much, Mr. Maverick Tamayo. A pleasant morning or noon to everyone na kasi it's almost 12 noon. So, pleasant morning or noon to our SINSM participants and to the faculty members of our Department of Biology, led by our department chair, Professor Roland Hipol. And with us also today are our research assistants of our Department of Biology. First of all, I would like to congratulate and thank our brilliant and talented speakers for this year's SINSM. These are junior faculty members of our department, but as you have seen and heard them in their lectures in the past 2.5 days, I'm sure you will agree with me that they are very competent, efficient, and intelligent teachers in their own right, and our department is very proud of them. So, special shout out and congratulations po sa kanilang lahat. Mag-congratulate naman po tayo dun sa ating mga reactions para sa ating mga speakers. So, special shout out to Dr. Esther Joy Saho, who shared with us her expertise on cell biology. She also covered the demo on cell culture and different model systems or bioassay techniques that one could use in the laboratory. Then we have also Dr. Madison Munar, who shared with us his expertise in molecular biology, and he discussed open source bioinformatics platforms like BLAST and phylogenetic analysis. And last but not the least, this morning, you have heard Mr. Jenny Ray Pagaduan, who discussed teaching genetics in an online setup, which included the lab demo that he shared with all of us. I hope that you were able to pick up or learn many updates in these fields of cell and molecular biology, as well as genetics that you could use in your respective online classes. So we would like to thank to our equally able, young, and energetic faculty members, plus the research assistants of our department who acted as facilitators, moderators, MCs, and they were in charge of behind the scene preparations of the materials and activities that were shared with you in the past 2 days. So let's also give them a hand. Special shout out din po sa kanila. For the last 2 days, you, our dear participants, have been busy, hooked onto your devices, whether it's a laptop, a desktop, or iPads, or maybe some of you used your cell phones, listening and viewing what our faculty members have shared on topics in cell and molecular biology, as well as in genetics. Thank you for being with us these past 2.5 days. Thank you too that despite our situation now, you found time to be with us in this virtual or online SINSM. Thank you very much for supporting our 2021 SINSM and actively participating in the discussions. The past 2 years have been very challenging for all of us because of this current pandemic. In fact, last year at the onset of this pandemic, we were not able to offer or deliver this SINSM because just like you, we were busy preparing our course packs and modules for our remote learning. However, this year, when we have adjusted to our situation, our Department of Biology and the whole College of Science here at UP Baguio made sure that we will stick to our mandate to continuously give current updates in the different fields of science and mathematics to our tertiary and secondary teachers in the region, despite all these challenges that we are experiencing right now. We hope that with these workshops and theoretical concepts that you have learned in the past 2 days, it will help you prepare for your respective online classes. These are very basic but powerful, practical, and helpful tools that you could actually use in your classes so that your students can also be updated and now be knowledgeable on all of these topics. So with this, I am formally closing our Department of Biology SINSM 2021. We are very grateful that despite our situation now, you were able to join us in this online SINSM. Congratulations to everyone, because after this, you will be getting your certificate of participation in our 2021 SINSM. To those of you who regularly participate and travel to Baguio City, we miss you here in our UP Baguio campus, and I'm sure kayo din you miss traveling to Baguio City. Kasi after your participation in our SINSM, kasabay na doon yung pamamasyal after at pamimili ng gulay sa palengke ng Baguio. I'm sure you miss all of those activities. So let us all pray that our pandemic will be over soon, and now that the COVID-19 situation, and pray that the COVID-19 situation will improve in the next few days to come. And when you have the opportunity to have the vaccine, please grab the opportunity because you will be protected. So stay safe and take care, and God bless everyone. Maraming salamat.
Thank you very much po, Ma'am Rose, for those warm words. Sobrang happy and fulfilling po na hindi nakaka-impart po tayo ng knowledge po sa ating mga educators. Ayan po, maraming salamat po sa tiyaga ng lahat ng umattend at ganoon na rin po sa expertise po ng ating resource speakers at sa lahat po ng nag-asikaso para maging maganda at maayos at matapos po ang ating SINSM 2021. Can I please request po lahat ng members ng SINSM committee sa Bio Department to please open their video? Because we would like to formally and award this bouquet to Ma'am Rose.
Wow, surprise yan ha?
Opo, ayan po. Opo.
Bakit may
And,
pa-bouquet?
Ma'am Ascon po, congratulatory po ng department po being selected po for our Vice Chancellor for Academic Affairs and for all your efforts, ma'am, sa department po. Ito po.
Wow, thank you very much.
Thank you, ma'am. Okay, sige. Kinuha po ni Sir Ronnie.
Pwede
Okay,
po ba tayo class picture?
Mag-class picture daw.
Okay. Ayun po, may hinahabol daw po si Sir Don. At this juncture, may I please ask everyone to please turn on their video po for our photo opportunity with VCAA Rose. Thank you po.
Ito, participants.
Okay. Alright. Sige, ako na lang po mag-screen cap.
Smile.
Smile. 1, 2, 3. Okay, next po tayo. Screen 2. Dami po nating participants. 1, 2, 3. Okay po. Screen 3. 1, 2, 3. And last, screen 4. 1, 2, 3. Ayun. Okay. Maraming salamat po. Okay, may I please ask the SINSM Tech Committee to please share to us the screen for our awarding of the certificate for our STEAM members, STEAM resource speakers for this year's SINSM. To read, Summer Institute in the Natural Sciences and Mathematics, College of Science, University of the Philippines, Baguio, awards the Certificate of Appreciation presented to Professor Maria Easter Joy Saho for being keynote speaker during the 26th Summer Institute in the Natural Sciences and Mathematics with the theme Upgrading Senior High School Science and Mathematics Education, Content and Competency, Part 1, via online conference platforms held on May 13, 2021. Signed, Dr. Rizabel Adawe, Chair of the Summer Institute in the Natural Sciences and Mathematics, and also signed by our Dean, Dr. Dim Fernando Lasco Javier. Same content is awarded for Professor Madison Timonar and also for Sir Genoray Pagadua. Let's give them a virtual clap. And also to Professor Romeo M. Dizon for steering the capsule proposal talk. Also for Sir Ronnie J. Kalugay. To Ma'am Rosel Amor Andong. To Professor Meiji Bagangao. And to Miss Kimberly Pagluwayan. Meron pa po ba? Ayun. So, yun na po ang ating last. Okay. So hosting the SINSM in an online platform has been a challenge and an unforgettable experience for everyone, especially for the steering committee. On behalf of the Department of Biology, we are very thankful for your active and consistent participation throughout the 3-day seminar. Tuwang-tuwa po kami since day 1 na very participative po ng mga ma'am sir natin. Ayan, na nagre-recite, na nag-share at nakikipag-engage po sa mga discussion ng ating mga resource speaker. It proved po talaga no na though nasa online setup po tayo, we were able to weaponize successfully online platform to bridge learning. Mabuhay po tayong lahat ng mga guro na nagsasakripisyo para sa kabataan, para sa ikauunlad ng ating bayan. Huwag po natin kalimutan ng evaluations po for the SINSM. We will keep in touch po regarding sa release ng inyong mga certificates. Kita-kita po tayong muli hanggang sa ika-27 SINSM po ay magkita po tayo. Maraming salamat. Diyos te agnina, ken mang tarabay kada tayo amin. God bless us all and happy lunch po sa lahat. Thank you, thank you.
Thank you, sir.
Thank you, sir.
Thank you po.
Thank you po sa
Thank you
lahat.
po.
Salamat
Thank
po sa
you so
lahat.
much.
Thank you.
Ayan po, for public and private HEI, meron pong invitation here to join an online lecture training with the Philippine Society for Cell Biology and in partnership with St. Louis University School of Natural Sciences. This is for Cordillera Administrative Region po, public and private HEI.